Hello students, welcome to the last of the lectures on chapter 2. This will be about nucleic acids, section 2.11. Let me go back. So nucleic acids are things like DNA, RNA, and ATP. We're going to focus on DNA and RNA first. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. All nucleic acids will contain carbon, oxygen, hydrogen, nitrogen, and phosphate. These are polymers. Their monomer subunit is what we call a nucleotide, which has a nitrogenous base, a pentose sugar, and a phosphate group. Let's see what that looks like. So DNA, deoxyribose, it uses 4 nitrogenous bases, adenine, guanine, cytosine, and thymine, and they base pair with each other. It's called a complementary kind of pairing. A will always pair with T, G will always pair with C, and vice versa. So C always pairs with G, and so on and so forth. The pentose sugar that is found in DNA is deoxyribose, hence deoxyribonucleic acid. This is considered a double helix. It's double-stranded. It is found in the cell's nucleus, and this is our everything. This is it. This is our genetic material. This is all of our instructions for protein synthesis. This is why I have brown eyes, some of y'all have blue eyes, and so on and so forth. This is the master blueprints for our body. Here you can see, here's the double helix. You can see here's the base pairing going on with an adenine nucleotide and a thymine nucleotide. So again, here's the phosphate group, here's the base, and then the deoxyribose sugar. And you can see that it base pairs A with T, T with A. And it goes like that all along the whole 2 strands, and that's how we end up with the double helix, by this base pairing that occurs. A with T, T with A, C with G, and G with C. Ribonucleic acid also has 4 bases. Like DNA, it has adenine, guanine, and cytosine, but it does not have thymine. Instead, it has uracil. Its pentose sugar is ribose instead of deoxyribose, and it is a single-stranded molecule. So it's not double-stranded like DNA. And RNA tends to function mostly outside the nucleus, whereas DNA is strictly inside the nucleus, and it's basically the workers. It's what kind of carries out protein synthesis. And there are 3 primary types of ribonucleic acid. We have messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. And we are going to talk about what all 3 of those things do in the process of protein synthesis in chapter 3. Since they are both nucleic acids and they have some similar structures, let's watch a little video kind of to compare and contrast the 2.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
So that gives you a little...
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
All right, the double helix of our DNA. It's our genetic material. We're going to talk a lot about DNA and RNA in Chapter 3. Again, to just kind of compare, there are different differences. RNA has the ribose sugar. DNA has the deoxyribose sugar. Instead of the thymine that we find in DNA, we have the uracil nucleotide. DNA is double-stranded, RNA is single-stranded. DNA functions primarily in the nucleus, and RNA functions primarily in the cytosol. The other of our nucleotides the nucleic acids that we're going to look at is something called ATP, or adenosine triphosphate. And what is incredibly important about ATP is that it is our energy molecule. It is usable by all of the body cells. It directly powers almost all the chemical reactions in our body. And when we break down our food to get energy that way, we convert it into this adenine-containing RNA compound, and that's essentially what ATP is. We have an adenine nucleotide and 2 additional phosphate groups, which gets us to adenosine triphosphate. So here's the adenosine molecule. It's an adenine nucleotide plus the ribose sugar, and then we attach various phosphate groups onto it. If we have 1 phosphate group, we have adenosine monophosphate. If we have 2 phosphate groups, we have adenosine diphosphate. And if we have 3 phosphate groups, what we have is adenosine triphosphate, or ATP. And what is significant about these phosphate groups, particularly this one here with the bond between this 3rd phosphate group and this 2nd phosphate group, is these bonds, these chemical bonds, have a ton of stored potential chemical energy. And when we break these bonds, we release that energy, and that is why ATP is our energy molecule, our energy currency. When we do snap off that last phosphate group, we call that phosphorylation. Essentially what we do is we take, let me go back here for a second, this 3rd phosphate group, and we break it off from the rest of the molecule, and we take this phosphate group and we transfer it to another molecule. We phosphorylate that other molecule. And by breaking the bond here between the phosphate group and what essentially becomes ADP, we have a ton of energy to do really useful things in the body, whether it's transporting things across cell membranes, whether it's contracting our muscle cells so that we can move, or maybe just some mechanical work. That's a type of mechanical work. Or maybe just making new molecules, using that energy to power other things in the body. So we have chemical work, mechanical work, and transport work. Practically everything in the body is going to use ATP as our energy currency. It is our main energy molecule. And that is the end of chapter 2.